Hello, everybody. We're back. The next session is about glycans. So our first speaker is Kiyoko Aoki-Kinoshita. Take it away.
Hi. Okay. Thank you. Thank you to the organizers for allowing us to speak here and have a whole session on glycans. I hope it's interesting for everyone. So I'll just go in. So glycans. Who has seen or anything about glycosylation anywhere in your data? Is it on? Oh. Hello. Is that better? Okay. So glycans, these chains of monosaccharides, and they're found on glycolipids, they're found on lipids, on proteins, and even free in the extracellular matrix. And they have lots of functions. We know that they modulate protein functions. They participate in cell-cell interactions. Viruses recognize them. There's so much information that is included in glycans, but they're often ignored. So our mission has been to try to organize this information and make it available, and to allow it to be accessible to other databases and to better understand biological functions through them. So just as a background, this is the symbol nomenclature for glycans, SNFG. So this is the table, it's the standardized table of monosaccharide symbols used to represent them. So we use these as the basic building blocks, and these represent all the mammalian monosaccharides. And there's rules for displaying and representing non-mammalian monosaccharides like that, those found in bacteria. So in order to better integrate glycan data, we've been developing Glytucan. This is a play on words. To means in Japanese sugar. And so when you talk Glytucan, you want to put sugars in a can. So it became toucan, but it's not a bird. The Japanese don't use there's no toucan in Japan. So you have the gly in the beginning, so it gives, glycans. But we do use a mascot. Anyway, so these the Glytucan IDs are used to provide unique identifiers for specific glycans. And the glycans don't need to be fully defined. Like these all have the anomeric configurations and everything known for this linkages. Glytucan IDs can also be assigned to monosaccharide compositions because oftentimes there's no sequencer for glycans. So mass spectrometry and other technologies are used, and they don't necessarily identify all the parts of the glycan structure. But they can all get different Glytucan IDs. Okay, and we are also part of the GlySpace Alliance, which is a three-partner alliance currently between Glycosmos in Japan, Glycosmos Expacy in Switzerland, and Glygen in the US. So the URLs here. There's a publication that kind of describes what each of the partners do, and our main goals are to really promote the usage of Glytucan IDs, promoting other standards for glycobiology information, protocols, metrics for data reliability and QC, consensuses for data filtering and annotation. And basically, if one of us goes away, we'll have some way to support each other and have a globally distributed data storage. So okay. So, but going back to the Glytucan data repository, now we have over 250,000 registrations, but at this time of last year, we had about this many, but there are over 35,000 nearly identical glycans. So what is this nearly identical glycans? We define it here as the same structure, but having different reducing ends. So the reducing end is where the glycan is attached to either the protein or the lipid, or it's been modified for experimental purposes. They get arrayed or they get opened up. They can have different anomeric configurations, etc. So this is, next. So, and there's also times when we don't know what the reducing end is. And so all these are given different Glytucan IDs because they're considered to be different glycan structures, especially to the glycochemists. So this is a specific example of how we have these 4 different Glytucan IDs for what look like the same structure, right? We have issues with our imaging display. There's actually 2 different structures where the ring type of this monosaccharide is pyranose, but we don't know what it is for this one, etc. So, and this is a problem because if we have someone searching for this kind of structure, they're expecting to get the N-glycan core with this ID. They actually get a number of different glycan IDs that look the same. They're nearly identical. And so this is an issue which we wanted to address. So yeah, basically, glycans are often, if in biological terms, we're most interested in just getting this, but we don't really care about the reducing end. But in the end, we'd have to go through searching for all the different structures that have this structure, and then incorporate it all and then decide which one we want to use. And it also hampers data integration as well. Which of these structures IDs do we use to represent this structure? So in order to address this, we have come up with we first looked at what these structures look like. So this is a works representation for this structure, and I don't want anyone to be able to understand what this is. It's just called works. Okay, it works. So the red parts are what is different. Basically, this first bracket represents this first monosaccharide, and we have different symbols to indicate the anomeric configuration or the ring connection, and these are all different variations of those. But this first one, we use the U representation, which basically refers to this entire group of monosaccharides. So this U residue is basically encompassing all the other reducing end variations to itself. And so it's possible to use, to select this one as the one that represents the entire group of monosaccharide or glycans. And so we're calling this the archetype glycan. So this archetype organization is what we came up with to select the one glycan with the U at the end as the archetype, and then link to the other glycans with the different anomeric configurations and reducing ends. And so in that way, we can organize all the data in Glytucan, whereas currently, these are all separately stored in Glytucan, but we can organize them into groups where the archetype connects all the different variations of itself. So by improving search, we can then allow users to just search for archetypes only, for example, and then those will be linked to the other variations of glycans, helping to improve search and make things more efficient. So, and it also helps to link data resources. So we have links with PubChem and Reactome and many other resources right now, and they may be using these different variations of the glycans, but whereas we can then use the archetype ID instead to really better integrate all these different resources. So we also use RDF to store everything, all our data in the semantic web. The main part that I'd like to focus on here is the saccharide concept. So we have the saccharide is what the class that we use to represent all the different glycans. They're linked to citations and evidence and source information through the other classes. But here we've now added a new archetype class as a subclass of saccharides. And so this allows us to use two bidirectional predicates to link the archetype with the individual glycans. So in our workflow to organize all this information, we have these 4 steps. We had to go through and extract all the different glycans from Glytucan, generate, so because we know we just need the U structure for the monosaccharide and the reducing end. We generate the archetype glycan or works string for each of these, and then collate all the archetype groups into based on the archetype structure, and then load this into generate the RDF and load it into our database, and then create the user interface. And as a result, we are at the time, this was a while ago, but we had this many glycans, and over almost half of them were we could organize into archetype groups. There are a few glycans that could not be made for which archetypes could not be generated. And these are strange ones, whereas, for example, this one is a monosaccharide and it is labeled as just a sugar. So we don't know anything about it, but we know there's a sugar in it. So there's no, even the U concept cannot be applied to this kind of glycan. So there are a few of these. Also, monosaccharide compositions cannot, at this point, could not be generated. We are figuring out ways to do this though. And then there are structures that are repeating units. So they're just, so this monosaccharide is attached to another glucose, and then in N repeats. So we have to come up with a way to take out the first part of it, make it into a single non-repeating unit, make that U, and then link it to the repeating. But anyway, yeah. So these are things that we need to work on. So these are not handled right now as archetypes, but we have ideas to handle them in the future. So, and we also need to create a workflow to automate this. Whenever a user registers a new glycan structure, we have to go through the generation of the archetype, identify whether if it exists or not already in Glycan. If not, we have to generate it, register it, and then update the archetype groups, updating the RDF, and then going through and automating this whole process. And so this system is currently being developed and should be available soon. But as an initial test, we did confirm that we could generate this many archetypes, and maybe 10% could not be generated based on depending because of these different reasons. So, and we're also working on the user interface. This is a very preliminary preview. It should be better in the future. You can list up all the glycans in Glycan, and you have this little radio buttons to filter them by all, and then we'll be adding this option to select only the archetypes. So this will allow it will also just displayed in the user interface, right? And then by clicking on each glycan, we'll have this is the original interface for the glycan entry page for this glycan structure, but we will implement this additional link to the archetype glycan if it's not an archetype already. And then if it is a glycan archetype, it will list all the other glycans in that family, and along with the other information that indicates why it's not an archetype itself. So, and then these, of course, these will be linked to their entry pages in the end. So we are working on, okay, as a summary, we've figured out a way to organize all these different nearly identical glycans in Glycan, and it was able to condense 65% of the original system. And the workflow has been developed. Now we're just trying to get it into RDF and updating the user interface for our next release of version 4, which hopefully will come out in the next couple of months. And that's it. The funding support, I'd like to acknowledge JST and BDC, and also NIH as a common fund. Tom Nasding was the main person who organized all this archetype information, along with Akihiro Fujita, who is at Nagoya University now, along with my programmers and then the Noguchi Institute, who have been developing the works format. Okay, thank you for your attention.
We have time for one question.
Thanks. This looks really interesting. I can't help but look at this and maybe it's just because this is the way I see the world. This looks like you're building an ontology.
Yes.
Okay. So have you thought about what kind of standard ontology building tools might help you to do this efficiently and in a maintainable manner?
What do you mean?
So there are lots of groups who built tools, among them gene ontology for constructing and managing ontologies and
yes.
Yeah, and have you thought about this and maybe employing some of those tools and infrastructures? Yeah.
Next speaker after Raja will be yeah. Well, he's not talking about it, but he has developed an ontology called GNOME, which is exactly what you're saying.
This is an ontology.
Which talk into the microphone. Is text space doesn't help us here. The text is not human interpretable. Pictures are. And so having a graphical way to represent the ontology is absolutely important.
Right, but so you're saying you're not giving it a text name.
The
You're
text
giving it
names
an
are not
image name.
useful for understanding the relationship,
Mhm.
only
That's-
the visual.
I think that, I think that's probably okay, because at the core of the ontology is an identifier.
Yes.
And if a text label is not helpful, that's perfectly okay.
Right.
Yeah.
Yeah.
Alright, anyway.
But
yeah,
yeah,
that's
there's
great.
lots of work going on to, make it more accessible, using these ontologies and labels and, Thanks.
Okay, thank you very much.
Thank you.
So I want to thank the organizers. This has been a fantastic meeting, and it's one of my favorite conferences that I like to go to. So I'm going to talk about biomarkers. And I have just one slide at the end because glycans can also be biomarkers, but I want to introduce the concept of biomarkers and how at least we are looking at them. So luckily, so I started thinking of biomarkers quite some time back with actually a few people who are in the audience. And over the years, at least I came to the conclusion that it's not really easily describable. I talk to somebody and they will say, okay, this is a biomarker. Then you start going a little bit deeper and you realize that unless we really understand what we are talking about, we cannot really communicate information, cannot connect it to other resources and so on. But luckily, the NIH, FDA, the FDA NIH biomarker working group, they have been working for some time. And if you have not seen this picture, this is a really interesting picture actually. And it's at a very high level classification of biomarkers. Many of your resources, you have content related to biomarkers, but it may not be organized in a way that is easily extractable, at least for a project that I'll be talking about, which is the biomarker knowledge base. So BEST biomarkers, BEST stands for biomarkers, endpoints, other tools. You can have diagnostic biomarkers, response biomarkers, prognostic, safety, risk, predictive, and monitoring. And if you talk about FDA or any other agency who is trying to use biomarkers in submissions, this is absolutely important to understand what type of biomarker you're talking about. If you're writing a paper, it might not be that important. As long as you can explain in your abstract and your paper what you're talking about, you might actually sometimes use a different terminology to talk about the biomarker, but that's okay because you're explaining with a lot of other pieces of information. But when you're trying to model data, it's really important that we have the right terminology. So, and these have definitions. There's actually a book, bookshelf, NCBI, you can take a look at it. So that's where we started. But then came the most interesting part, at least in our understanding of biomarkers. Most of the resources, if not all of the resources that we have looked at were not biomarker specific. When you mention that it's a biomarker, you mention the entity. You say EGFR is a biomarker. Well, EGFR is not a biomarker. You have EGFR, I have EGFR. So mutation, the change over expression of EGFR or lower expression or a mutation in a particular position can be a biomarker. And I think that was the key. So when you have a biomarker, is the alteration that we are interested in. Of course, the assessed biomarker entity, again, majority of the groups here who are talking about databases or knowledge bases, you have entity IDs. Suddenly we can start mapping to things. And when you start mapping those things, the green items are the ones we absolutely need. We need a condition, a disease or a condition. There's a slight difference, exposure agent. I think there was a question earlier about exposure. So you also have biomarkers if you have been exposed to something, right? So you can have those IDs here. If you have these green items, then it becomes relatively easy to connect to a bunch of other things, Uberon, specimen, LOINC. If you're in the medical informatics space, when you go get a blood report, there's ID. So LOINC is one of those IDs. You have evidence and so on. Now, that was an overly simplistic view of what I said. So I have a great collaborator. He's a medical informatics. He also has an MD. And I started talking to him. He kept down the phone. I was on the phone call with him. He's from our university. And he sent me this picture. He just did it over the evening. And next morning, I get this picture. And if you look at it, it is the sensitivity, specificity, performance of a test. So his main thing was all biomarkers you're talking about, they're all tests, right? And if you think about it, yeah, you're right. I mean, in a clinical space, it's a test. And then you have these tests, these biomarkers that talk about how critical or severe, moderate, mild, asymptomatic, test properties, disease properties, and it is still like not the full picture. So I'm not saying that we will be able to solve everything by doing a biomarker data model, but yes, we can start thinking about a lot of these things, and a lot of these things can end up as metadata maybe. Ontology, I have to put ontology slide. We don't have ontology, which is RoboFoundry, but there was a talk earlier on by Hande, and she talked about the ontology that she has been working. We are working together. This is something we are working with Darren Natali, and I think Lynn is also here. You know, she and us, we have talked quite a bit. So we published this paper in the International Conference for Biomedical Ontologies. I like the title, Analysis of Biomarker Data Towards Development. So you see, you know, we don't want to say that we have developed anything. It's towards development. And it's really interesting because we wanted to know even the entity types at the higher level nodes are the ones we are really interested in capturing so that we can do things. Then COVID happened. And like many people, actually, COVID was a very interesting case. There were 0 biomarkers for COVID-19 at a certain date point, right? Because the disease didn't exist. There's 0. And then every week, every month, papers started coming out of biomarkers for COVID-19. It was really interesting to see. And then we had lots of people sitting at home, wet lab research scientists, students. They said, can I do something in bioinformatics? So I said, let's curate the COVID-19 biomarkers. And that was a really interesting experience in real-time biocuration as data is coming out. And we didn't have the baggage of what was known before, right, about COVID-19 biomarkers. So we did publish this paper, and we also started talking to clinicians. And it was also interesting. The clinician in our hospital said, we are using this biomarker to decide who's going to end up in the critical care unit. And I said, are you talking to other hospitals? You know, no, the answer was no. So because these were hospital-driven biomarker usage, there was no database, there was nothing. So it was really, really interesting. And again, if you see this little bit of this curve, you know, the risk biomarkers sit here, diagnostic, and then, you know, different biomarkers are used at different points in a disease state. And also, you need to also, or at least we started appreciating a COVID-19, let's say interleukin-6. Okay, there was a paper saying interleukin-6 can be a really important marker. If a person has cancer or some other disease, that biomarker may not be at the same level as somebody who is healthy or a different age. And that is really, really important. How do you then look at people who have comorbidities? How do you look at it? How do you model it? How do you even understand what's going on? And this figure, so all of you have had blood reports that you see, right? And then there will be, hopefully all of it is in green, maybe once in a while for those of us who are getting older, something will be a little bit not green. And it will tell that, oh, you are not within the range. Who decided that range? Yes, there is a lot of work that has gone in. And it's also clear that range is not always the last word, right? Your clinician who has seen lots of patients will tell you, don't worry about it, just, run 2 extra miles every day or something. So it's still very much dependent on, what the clinician thinks. So what we started doing is that we, many of you may already have been thinking about it, get access to clinical records. So Cerner or Epic in the United States, there are similar things in other countries. So you can start asking questions. If I map the biomarkers really well, then I will be able to know what are the ranges of certain biomarkers in the population, even at a very, very granular level. So we're working with a couple of groups who are collecting data like this, and we're mapping our biomarkers to the clinical data, and we are slowly trying to understand for those biomarkers which are already tests and are in the clinic, what's going on. But having said that, we are not just interested in biomarkers that has gotten FDA approval and in the clinic. We are collecting biomarkers even from publications which are not in the clinic yet, because it's supposed to be a discovery, and future work also should be can use this type of information. So we launched literally last week. So we were for a long time, we were on a testing phase, and we have biomarkerkb.org. So you can do biomarker search. Our infrastructure is hopefully is very clear. We have a portal, which is biomarkerkb.org. And then from the portal, you can do searches. You can still read about and like classical, and then the statistics of what's in there right now. And if we don't have any biomarkers that you think your resource has, please talk to us, and we will be more than happy to talk with you to see how best we can get it in. Sometimes it looks like we need to use NLP and other methods to better extract the data, because remember I talked about the change. So maybe if your resource has just this gene is a biomarker, this glycan is a biomarker, or this metabolite, we will not be able to use it. We want to know the change and the disease or the condition associated with it. And sometimes that is in free text in your resource maybe. So we need to work with you to figure out, but we are trying to come up with some ways to generalize extraction from free text with collaboration with Caltech, our collaborator, and JPL. So you get a table like this, and we have biomarker IDs. Yes, we do need a different ID format, because you can't really use the entity IDs to show what your biomarker is. We also need to have almost similar to canonical versus isoform concept. We need to connect certain types of things together within the biomarker space so that people can explore things and understand how these things are all connected to each other. And these are like filtering mechanisms. You can say, you can do some search, you can then filter out things to get to what you need. And there are a lot of talks about knowledge graphs, and you can already start thinking, right? If you have a biomarker-centric view of some entity, biomolecular entity, then you can connect them with information from a lot of other resources. So we have a collaboration for a common fund project, and with Dr. Avi Mayan's lab and Dion Taylor and others, where they are building knowledge graphs with information from a lot of resources, and some of the resources that you all build. And we then add our biomarkers to this graph, and we can start asking questions. So there is already a web page that you can try out. We are trying to build out more detailed ways of more comprehensive ways of interacting with these knowledge graphs. We are using Neo4j cipher queries, yes, but there are maybe other ways one can download and use these type of graphs. So we also try to organize our resource in very clear ways, hopefully. So we have biomarkerkb.org, but then we have data.biomarkerkb.org. So there you can download all of the data sets that goes into the biomarker portal. And all of our data, anything that comes in is described in biocompute objects. So biocompute object, you can describe, you can have scripts, you can have information of how the data was collected and harmonized and so on. Biocompute object is the IEEE standard. It's used by FDA centers when you are submitting stuff to FDA for, earlier, what would happen is that people will say, I use this script to run my analysis, and here are the results. So biocompute goes beyond that, and this is a FDA-funded project where we have been working closely with pharma and others to describe what was done. And of course, we have the APIs to get the data. Everything is CC by 4.0 license, so you can use that. And we have another thing that we are doing. I don't know if how others are using the wiki, but we use MediaWiki. So we can quickly update and document. Every curator can update and document very quickly, very fast. So this wiki provides additional information that we constantly don't need to update our portal, but we have links from the portal to the wiki. I am one of the PIs. Mike is here. He's the other PI for Glycan. You have heard about glycan in the earlier talk by Kiyoko. So biomarkers are also integrated into glycan. So this is an example. If in your knowledge base, you want to have another section about biomarkers, and if you are the same entity, like let's say you are KB, so then it will be, let's say, whatever KB entity you have. If you are a protein-based resource, then you can say this protein has these biomarkers associated with it. So you can do searches in there similar to the ones that I was showing, but it also connects to other information on glycans and proteins within glycan. So this is a high level. Why we are doing this? At least why my group is interested. So we have this ARPA-H project, and this is an example. So in my title I have the word translational somewhere in there. So I have one slide on that. So we have the hospital data from Epic. So Epic Chronicles goes here. We have a de-identification tool, which the EHR, EMR data goes here, but we also collect data from other in-house omic. We are working with advocacy group, breast cancer, so advocacy group, where they have also shared data with us. So all of this data gets harmonized, and I don't need to explain what harmonized means. It's the right IDs, right? So that it can talk to knowledge bases on the right side. And this data, we are working with Georgetown University, Cornell, Embel EBI, Vanderbilt, NCI, and Kaiser Permanente to allow people to search from our hospital data, their hospital data, all is de-identified. It's an ambitious project. We are on the 6th month of this project, and BiomarkerKB also we can enhance when people have data from their clinics. They can use this kind of data to better understand. The clinicians can better understand how to interpret the data as more and more data becomes available. So that's it. That's my last slide. This is acknowledgement. It started as a biomarker partnership. There were previous projects which led to this, and right now we are in the BiomarkerKB NIHU 24 mechanism. Thank you, everybody.
We can have one very quick question.
Thank you. Great talk, and it's a very needed resource. As you pointed out, context is a lot because everything is a biomarker. So contextually, everything can be a biomarker, right? So putting it contextually would be interesting. So for example, I looked up PTH as a biomarker, parathyroid hormone as a biomarker. And from what I've seen, the information that you currently have is regarding mutations in PTH, because that's some sort of cancer indicator. But another function of PTH that is clinically used very often is, for example, an indicator of calcium disorders in the body. So my question, I guess, is once you have this one biomarker, PTH, that can indicate different things in different contexts, even just PTH by itself. Hyperparathyroidism is another issue, but it can be primary or secondary, right? Things like that. How would you capture that?
I'm hoping that some of you all will help me to capture that. Yeah, but at a very high level, I'm hoping that if any biomarker is already being used in the clinic, that capturing could happen potentially through mining of data from electronic medical records. The other thing is that that information, let's say, is going through the approval process or clinical trial. So we are in the first phase. So this is our 6th month right now. So we have collected a lot of data, and we want to continue collecting till the end of the year. And ideas about how to add additional knowledge, contextual knowledge, which is really, really important, has to be happening. So that year 2, that's what our main goal is. But we've already started collecting. So one graph, little figure I showed. So we are trying to collect this type of information as much as possible. We also have a consultant who is working on text mining from publications. So we want to kind of add all of this information. How to present it is a challenge. So I hope that, and that's why I'm here, trying to learn how others are trying to work on these kind of
Okay, just pay attention, because having worked with clinicians on these things, certain biomarkers can have a wide range of applications, and the wrong use can be the wrong indicator.
Absolutely.
So, yeah.
Absolutely, yeah.
All right, thank you, Raja. All right, for our last talk of day 2 is Nathan Edwards. 2 notes before you start. We are going to take a group photo directly following this talk on the stairs, so do not run away. We're all gonna, Sophia's gonna tell you exactly where we're going.
Thank you. I think this might go somewhere up the leaderboard for longest title of the day. Just a tiny bit of context. The idea of inferring specificity or function from gene expression data or even single cell data is not new to this community. The reason this is, I think, is interesting is that glycans as a substrate for functional annotation have almost nothing that isn't in somebody's head. How can we take advantage of the sorts of tools that we know how to use from our gene expression data? How can we leverage them to try to say something about glycans as fundamental objects that have function in the cell and in our various pathways? Quick introduction for those of you who are not familiar with glycans. I wasn't until a few years ago, so I've learned all of this on the fly. Unlike our proteins and our other sort of molecular entities that we're used to working on, this is an enzyme-driven synthesis process. There is no template on the genome that we can use to understand what glycans are made and what to expect in any particular cellular or tissue context. Glycans are made by the sequential application of enzymes in the ER and the Golgi, gradually adding additional monosaccharides onto the structures. What I like to say is that while we know the toolbox, we know the enzymes which together build our structures, we don't know how those enzymes are going to be used in any particular circumstance. We have the toolbox, but that toolbox could be used to build a birdhouse or a bookshelf. And we don't know which of those is going to get built in any specific case or as a general rule. One of the projects I work on for Glycan is the Glycotree Sandbox. This originated with Will York at UGA, and it involves a sort of computational pipeline for taking our glycan structures and annotating them with the enzymes that are responsible for attaching each of the monosaccharides on those structures. Perhaps counterintuitively or not obviously, there's a huge amount of redundancy in the transferases which build up these glycan structures. This monosaccharide here on the end of the antenna, there are 6 different enzymes which are responsible for the addition of that monosaccharide with this specific linkage to this specific monosaccharide at its connection point. And we don't know a lot about when these different enzymes are used in which cellular context. We do have a clue, though, that changes or loss of these enzymes can be particularly deleterious. The congenital disorders of glycosylation are rare disorders in which specific glycosylation enzymes are lost or somehow compromised, and they lead to the loss of large swaths of potential glycans on our glycoproteins. So this is a change in one enzyme which results in a global change to the post-translational modifications on many, many proteins in many, many contexts. Most of these CDGs are highly deleterious, right? So you lose a huge amount of what the cell needs in order to be functional, but there is one in particular that can be resolved by diet. And that's the loss of the enzyme for the synthesis of mannose. And for a variety of reasons, which I don't have time to go into, this is one of the ones which we can manage by diet. However, the sort of implication here is that we have a phenotype, that's the disease, with we're connecting the phenotype with the fact that glycosylation has been globally modified by the loss of that enzyme. right? This is not just a small part of one pathway. This is the loss of this particular type of structure throughout the cell. So this asks the question, can we find evidence of this type of specificity? And if we look at tissue data, tissue gene expression data from GTEx, we can find evidence among these 6 enzymes responsible for attaching sialic acid. We can see some evidence of tissue-specific expression, suggesting that one or more of these enzymes is more strongly associated with whatever biological process is going on in that specific tissue. But we could also see it's a big mess. You know, really understanding what's going on. Tissue is a very, very crude way to assert some notion of function or some notion of behavior to a specific enzyme and thereby to the structures which it helps build. So the task here was to see whether or not we could use some notion of specificity with respect to single-cell RNA-seq data. Single-cell RNA-seq data is a lot more specific. The cell types have specific roles. And we can, what the hope is that we can use machine learning to understand whether or not the glycosylation enzymes expression are good at identifying the cells from a specific cell type or not. If they are, the assumption is that this represents some signal or association or functional role for those, for the glycan structures which that enzyme is responsible for building. So here I'm using the GTEx single-cell RNA-seq data, which has 8 tissues, 36 cell types, and 75 different tissue cell type pairs. We have 108 glycotransferases from the Glycan sandbox, which I maintain. And then for each of those pairings of glycosyltransferases and cell types, I'm going to build a simple logistic regression model and see whether it does a good job of picking out the cell type that it's supposed to be interested in. One of the problems with this is that I don't have any great null model for understanding when this is performing well or perhaps surprisingly well. Turns out that some cell types are incredibly easy to classify with any gene you choose. So we had to build an empirical p-value model based on randomly choosing genes and seeing how well that could pick out the cells as well. Here's an example of one of the sorts of things that we can find. The blue bar here represents a notion of the p-value. The green line represents a crude notion of multiple test significance, approximating approximately 50,000 tests. The red dots represent the percent recall at identifying the specific cell types. You can see that this particular gene responsible for attaching the sulfate to the GalNAc residue is very specifically found or very usefully associated with these 2 cell types, which otherwise don't seem to have that much in common. We can also see 2 others down here, which are not significant, even though one of them has quite a strong recall. As you can see, there's not a huge amount of chaff here. We're doing a good job of eliminating things which just do not seem to have good significance. It turns out that this enzyme is highly specific to this specific mucus cell type out of all of the cell types which were observed in the single cell data for esophagus mucosa. Again, this is potentially hypothesis generating, but certainly not conclusive at this point. Here's another enzyme which shows strong cell type specificity. It's one of the enzymes for attaching the sialic acid on the end of this structure. It has one particular cell type that's very, very significant, endothelial cells in lymph nodes. And we can see that in this case, it's actually significant when we group all of the lymphatic endothelial cells, regardless of the tissue, whereas the previous one was tissue specific and was not, did not have such a general utility. But the most surprising thing when I was pulling this work together was that when I started to look at pairs of enzymes, which together added multiple monosaccharides to these structures, either an O-glycan structure or a N-glycan structure, the addition of the O-glycan core here was actually more significant than the ones found on the N-glycan structures, which suggests that this specific enzyme association is tightly coupled to O-glycosylation. And we actually have a glycan motif, which has a name, TN antigen, which represents this specific structure implied by this highly significant association. I've timed things almost perfectly. Here's some quick acknowledgements. Abdullah Abdul in my lab worked on the first version of this work. The Glygen team has been instrumental here, both as a sounding board, and Mike for suggesting this as something which might have some juice, and I should try and get a grant to work on it. And then the Common Fund Data Ecosystem Grant and the Glygen grant, which funds my time on this effort. Happy to take questions if you have them.
Thank you. We have time for several questions.
Everybody needs coffee and sugar.
I'm curious about your graphics. I downloaded them. They're SVGs. How are those assembled? Where do they come from in the sandbox?
Do you mean the glycan structure
Yes.
diagrams?
Yes.
These ones are generated in JavaScript as part of this site. However, I do maintain a similar infrastructure for generating SVG images from descriptors of glycans. So that might be a resource that you would find useful.
Yeah, I'd like to talk to you later.
Sure. Yes.
I love the pairwise. How far out can you go? 4, 5, 6, 7, 8? What kind of profile can you generate in terms of multiple transfers?
Yeah, it's a huge problem because the combinatorics of doing pairs led to about 50,000 tests. Once we do all those combinatorics, which really provides bruising multiple test correction problems. Particularly since we're using an empirical p-value thing here, which involves sampling, and all of that costs time and effort. So the question is, I'm a little bit scared to go too much further than pairs. I was substantially surprised that we got this O-glycosylation result here. I was absolutely not expecting it. The fact that monosaccharide is attached at the O-glycan core, and that's a different set of enzymes, and that led to that specific observation that we have more significance for the O-glycan version of that. That was unexpected and more information than I expected to get out of that experiment. So the short answer is I don't know. There is some degree of limitation we can do it, but you can see that even with this one on the left, we're talking 20 odd times 6. So that's already a big number of trials just for the possibilities of assembling those two monosaccharides together. I don't know there's any great solution to that, but it absolutely something I'd like to try to do. There's another version of this which I didn't have time to implement, and that is that sometimes the addition of a monosaccharide to its base monosaccharide happens as a fork rather than as a series. And that's another version of pairwise that I could certainly recover from existing structures. It's possible there's some signal there too.
All right, one more round of applause for the speakers and yeah, for everybody.
All right. Well, that ends our talks for day 2. They were all fantastic. Once again, thank you all for being here.